Hi. Good evening, everyone. We will be just today examining a particular concept in biology called mutation. This word is not new to many people. However, we better try to know what is the meaning which I feel is correct based upon my experience of teaching. Now, mutation refers to the errors or the mistakes that happens in a cell during replication of the DNA, or it might happen at the mitosis, or it may happen at the meiosis, or it may happen during any of the cell activity when the cell is exposed to radiation, or anything of that sort might happen. That means it will be an error or a mistake in the DNA itself, in the genetic material itself. And that could happen at a very small level, micro level, especially at the DNA sequence level, or it might happen at a larger level like a chromosome. So this is one possibility. Similarly, it can happen by means of a mutagens. That means it is induced due to certain chemicals, environmental changes. All these things might induce the mutation in the organism. It might happen naturally. Now, naturally, mutations have been happening, and it is still going on. One example we can give would be the neck of the giraffe. By the use and disuse theory of that comes up in the natural evolution, it clearly indicates at some part of the time giraffe had a very small neck due to its nature, due to its length of the body, and being a herbivorous animal, it went on feeding on the trees. Therefore, some of the genes got modified. This is the theory, use and disuse theory. And similarly, there are examples in the biology. There will be sometimes a two-headed snake will appear. So mutations are happening in nature also without any mutagens. So we have to know only one thing, that is mutation refers to a error or a mistake that happens in the cell or in the cell cycle, either on the DNA or on the chromosomal level. It's possible. And these mistakes will result in the variations. The phenotype of the organism will change. There may be a result that the changes may be in the structural proteins. Changes may be there in the functional part of the proteins. And these changes which happen are due to or are possible that they will be inheritable. It can be passed on from one generation to other generation. Sometimes it may be fatal. Mutation may be fatal. And sometimes it leads to malfunctions of the entire organism itself. So these are all the various types of things that happens in the living system, especially in the organisms. When we say organisms, we talk about the plants and also animals. Plants and animals, and also this can happen in viruses. Today we have been witnessing a scene that this COVID-19 is changing its structure and function in attacking in about 20 days or 25 days. So that kind of mutation is happening. So these things are possible. So we have to know these things in a very simple way. Now, to come back to the point of what we are talking, change or a mistake in the genetic material. So we will now come to genetic material. So it may be an alteration in the nucleotides sequence. So you know very well that the genetic characters or the hereditary characters has passed on from one generation to other thing based upon the DNA sequences. If there is a change in the sequence of DNA, you know that DNA will contain a large number of things, and it contains bases like adenine, thymine, guanine, and cytosine. These are the 4 bases that make up the DNA of any organism. And that genome, or the total number of genes present in one species, is called a genome. So the genome entirely is made up of ATGC combination. Millions and millions may join together. If any change happens in this one, for example, if you take ATE and the copy of that will be DNA. If this is copied on the RNA, what we call adenosine, what we call the RNA molecule, and wherever you get adenine, thymine should come. Instead of that, you get the uracil. Wherever thymine is there, adenine comes in, and then guanine for C and G for C. So this will be the mRNA. By chance, any of these in the mRNA or in DNA or in DNA, anywhere a change happens to the base. For example, if this change happens, let us say for example, A, T, T, C, T, T, C, G, C, maybe for example in DNA. And in the natural process, this would certainly will produce adenosine, will become uracil, this will become adenine, this will become guanine, and this will become adenine, guanine, cytosine, and guanine. This is a normal RNA, mRNA. Let's imagine that way. Supposing there is a change in this one, and let's say this one, this will change due to radiation or something into A. When that changes to A, naturally this will become, now here what happens is, now when it becomes A, this should be T, instead of T it will be U. So now this particular change that happened on one sequence will be multiplied, will be amplified. That's the word, will be amplified in the gene expression. So any change, a minor change in the DNA will be amplified by the time it becomes a protein. So that also we will check up in the next session. So in this session, in a very general way, we will talk about that. So I just refer to you that the mutation refers to the errors or mistakes in the DNA that results in the variations, and the variations may be in the structural part of it, or it may be functional part of it, or it will be inheritable, or it may be even fatal, and malfunctions will be seen. And the mutations could be natural or it could be induced. In the induced mutation, there will be mutagens. This may be chemicals, environmental factors, natural radiations, isotopes. Any of these mutagens can cause a change in the genetic material. In the natural condition, the environmental condition, and depending upon the use and disuse of the organism, over a period of long time, it does not happen in 1 day or 2 days, over a period of long time, there will be large scale changes in the organism due to changes in the chromosome itself and also in the gene itself. So there will be a change, and that type of thing is called natural, that is called the spontaneous one. So now when we come to the organism level, at the organism level, DNA is the most important candidate that will bring any changes or that will bring any mutation. When the DNA gets into mRNA, at the time of replication, here, if there is an error, that results again in all these things. So these errors are possible. In the viruses, we have both RNA virus and DNA virus. If it is a RNA virus, a small change in the base of the RNA, one nucleotide is changed, then when the virus changes. We know how virus change very, very fast. This COVID, we have seen alpha, beta, gamma, delta, all these things are coming up, maybe because of the rapid changes or rapid on the velocity of the change in the virus. This is very, very fast. That will happen. Now, coming back to the types of mutations, normally we can think of two types of mutation. One is the point mutation, and second is the frame shift mutation. These are the two main types that we could study at the level of DNA, or at the level of the microscopic level. We can study these things. Point mutation, in that we have called silent mutation, then you have missense mutation, you have nonsense mutation. We will come to know what about these things, and we also have the frame shift. This is at the DNA level. If you take at the organismic level, at the organ level, for a long time level, it might happen at the chromosomal level. There may be, if this is a chromosome, a chromosome may have a new addition, that is insertion is possible. Then deletion, a chromosome, if it is like that, one part may be deleted, and substitution, maybe one chromosome may be added to the other chromosome, like this will be added. So these kind of changes will happen in a large scale. So that is also possible to bring in errors in the genetic material. So today what we do, we'll just take up one little example, and then go next time, then the next session, we will discuss these things in detail. Today we will just take up one thing called the missense point mutation. I will just give the one example. We have got what's called our normal hemoglobin produces, normal blood cells will produce a normal hemoglobin, and the red blood cells will be completely discoid in nature, like that. But if there is a change in the DNA, and sometimes the RBC will take this shape, that's called sickle cell. So RBC will not be round discoid, it becomes sickle cell-like structure. How this happens? It happens because only one of the codons in the mRNA, which codes for glutamine or glutamic acid, CAC, will change to GUG, where glutamic acid is replaced by valine. How this happens we'll study in the next class, and that will result in sickle cell anemia. That is a very simple example of the missense. Means the reading when the mRNA is there, and the ribosome starts reading that entire sequence. If it misreads for CAC, for glutamic acid, if it misreads as GUG, then it picks up valine, and that is enough to bring a change, the amplification change. That little change at the micro level amplifies at the organismic level, and the people of such thing will get what's called sickle cell anemia. It may be even fatal sometimes. So that is one example I have given you. One more example we'll study just to close the session today, called frame shift. Here, frame shift means we are talking of a large number of sequences, not one, or like here we have only one point we took, only one of them. In this case, we take a large chain of DNA and we will study. So this long chain may be able to produce a protein maybe a protein for nucleosome. Imagine that. a protein for nucleosome. It is producing. So what happens in this frame, if one of the codon in this ECU codon, we got 1, 2, 3, 4 codons are there. In the first codon, instead of uracil, that means I am talking of mRNA, instead of uracil, if we get, or if this is deleted, let's say this is deleted, if this is deleted, then the reading will continue. As the reading continues here by the mRNA, it reads C, C, G. So it becomes ECG, and then it further reads as ACU, ACU, and then it reads ACC, ACC. Then it comes to other one. So now the whole sequence has changed. The reading frame has changed. The entire meaning has changed. The entire structure of the DNA has changed by deleting 1. So deletion is one example we have taken frame shift. Similarly, there will be addition also possible in the frame shift. That will change the entire thing, and that little change will be amplified. That is the word used in the mutation. Amplification takes place at the gene expression level. We will stop at this part of the time, and tomorrow we will take up point mutation and frame shift mutation and something like insertion, deletion, substitution, with more examples that might appear for your examination. Just to recollect, we remember that mutation refers to a mistake or an error, either natural or due to induced, in which the DNA may change. One of the DNA nucleotide sequence may change, may be affected, or chromosome may be affected, which will be amplified at the time of gene expression. That's what's called the mutation. If you like the way in which I am talking to you, please try to subscribe to my channel. In every frame of my presentation, you will get a red sign, watermark. Please press that so that I'll get more viewers. Thank you very much.